Thank you everyone for joining the webinar today, and hope everyone's having a good week and a good new year so far. We have a webinar as part of our Emerging Trends in Digital Engineering series to kickstart the year. So the first area that we wanted to look at in Engineers Ireland was a focus on biomedical engineering and everything that's exciting and emerging in that industry at the moment. So we have three excellent speakers here with us today, which I'll get to in a moment. Just to cover off some housekeeping for those attending. First of all, if you have any questions at all for our speakers throughout the presentations, if you want to use the Q&A function on the Zoom toolbar there to ask any questions, and we'll leave time at the end for that, and hopefully have a bit of a discussion panel at the end with our three speakers. The second, so if there's any technical difficulties, if there's a problem with the sound or how anything's looking, if you want to direct your or highlight it to me in the chat function, and I'll look after that. And other than that, just to note that obviously this is a free online CPD webinar. So it counts for 1 hour of your CPD for the year, and with the CPD submission deadline looming next Friday, if you want to record this as one of your CPD hours, you just do so on your profile. So other than that, we'll get to it. So as I said, we have the three speakers today. So first of all, we have Dr. Jamie Concannon with his presentation on experimental and computational investigation of brain tissue damage. Then next up, we will have Dr. Andrew Daly, and he'll be looking at bioprinting human heart models from stem cells to functional tissues. And then lastly, Dr. Ellen Roach. So Ellen will be talking about augmenting heart function with device-based approaches. So firstly, I'll introduce Jamie Concannon. So Jamie's a principal investigator and program director for biomedical engineering at NUI Galway. And his research interests include medical imaging, finite element analysis, and bovine dystocia, and primary research area focuses on the biophysics of brain damage and remodeling. Jamie holds a PhD in soft tissue biomechanics and device interactions for vascular surgery, and his postdoctoral research focused on the development of a novel numerical technique for fluid structure interactions of the heart and the effect of boundary conditions on cell polarity. And Jamie's also received funding from the Irish Research Council and Enterprise Ireland, and awarded multiple national and international prizes. So with that great introduction, I'll pass it over to yourself, Jamie, if you want to share your screen.
Thanks, Steve. Can you see that there?
I think it's gone to the end of the presentation, maybe. Different slide.
How about now?
Lovely. Yeah, lovely.
Okay, great. Thanks a million.
Sorry, Jamie. It's just in the different display. If you go up to where display settings are, up at the top. See that? Yeah. There. Perfect. Yeah.
There we go. Yeah. Great. Okay, so thanks for the introduction. And yeah, so I'm going to be looking at our work on experimental and computational investigation of how brain tissue damages and heals and things like that. So the outline for my talk today is going to be kind of a brief introduction to the clinical problem. We're going to dive into the experimental testing that we've done on animal brain tissue. We're going to see how we've tried to start computationally modeling this, and then finally some ongoing and future work and some problems and future areas, that questions that we still have that we're starting to tackle. So just to start, my research group are primarily kind of focused on concussion, which is a form of mild TBI. And symptomatic recovery for concussion is generally in the range of 10 to 14 days. And what makes this a particularly difficult or interesting problem is that patients with concussion present with no visible damage. So if you see here, the image on the left is a major TBI. So you have these regions of hematoma and things like open skull fractures and all these things that we can see on MRI and CT. But this MRI on the right-hand side shows a patient that has a concussion, which looks perfectly normal. And this is a really, I suppose, interesting thing to look at then, because what is the mechanism of damage if we can't see it? It's not like a fracture that we can see on an X-ray and see how that fracture heals. So the clinical hypothesis or theory of what happens here is that it's the delamination at this gray and white matter boundary between the two, the outer gray cortex and the inner kind of white region of the brain. The only thing is that this is a theory and there is no scanning evidence for this. So it's something that hasn't been validated yet. And what makes this kind of particularly fascinating as a problem to research is that there's no visible damage. If you repeatedly get this kind of concussion or there's no visible damage, it leads to this aggressive neurodegeneration. So you see this on the left-hand side is a healthy brain, and on the right-hand side is this patient with advanced CTE or chronic traumatic encephalopathy. And you see this aggressive degeneration and atrophy of the brain tissue over time in a very, very short period of time. So to just, I suppose, motivate or set the scene, I wanted to kind of show you a case study. And this is the Miami Dolphins quarterback. You see he fell over there. It wasn't a very dramatic hit. You see his stumbles here in a second. This was one Sunday in September, I think it was 2022. You can see he hit the back of his head there. He was then cleared to play. So he passed the concussion protocol and he was cleared to play 4 days later. And you can see he fell backwards again, hit his head off the turf or off the grass on the posterior occipital lobe. And straight away he loses this upper body, upper extremity kind of motion. You've got this, what we call deep cortical posturing. It's what happens when you sever the brainstem in monkeys, and you see this kind of contortion of the fingers. So it leads to a really interesting problem in that this thing that we can't see is very aggressive in terms of neurodegeneration. And a fascinating autopsy study in 2017 showed that 110 of 111 players who play NFL had this CT on autopsy. So it's a kind of a rampant silent disease that we're trying to investigate the biomechanics of. So just to start, I suppose we tried to tackle this problem because obviously we had to kind of start with animals. So we went to our local abattoir and we got a lot of help from them in terms of dissection and being able to basically just take home regions of, well, whole brains, but whole heads really, and we dissected them ourselves in the lab here in Galway into different parts of the brain. So we've got the frontal, parietal, occipital and temporal lobes. We had over 30 sheep. And we then dissected these samples into gray matter, white matter, and interface samples. So the interface have both gray and white matter. So they're spanning across that kind of boundary. That's our, I suppose, primary focus to see if that clinical hypothesis is can be investigated or what comes out of it. And every one of these samples then was subjected to tension, compression, and shear testing. So these are the kind of the primary loading modes. This is a kind of an idealized kind of a GIF, but you can see as you hit the impact at the front or anywhere in the skull, that kind of wobbling of the brain is a mixture of tension, compression, and shear because of things like anatomical constraints and inertial effects and all these types of things. So we had to try and figure out, well, can we model this at least experimentally? First of all, can we start to take these samples and start to shear them in the lab? So we kind of have this schematic again. And what happens is gray matter is in purple here and white matter is in green. And when we shear them up to 50%, there's not that significant of a difference in the behavior. You've got stress on the y-axis here between the gray and the white matter. It's only when you start to do tension and compression. So we're going from the kind of the gray cube here to the kind of the elongated rectangle that they start to diverge. So again, at 50% tension, you'll see the purple, which is the gray matter, is that you've got this initial kind of a stiffness and then this softening behavior. It's like a neo-Hookean kind of strain softening that happens over time, or over strain, I should say. Whereas the white matter, which is in green here, has this kind of linear stiffness all the way up to 50% strain. So these are kind of the first kind of two things that we had looked at to try and see, do they behave differently? Because everyone kind of assumes brain tissue is just brain tissue, but we're showing that, like, well, it depends on where you look, first of all, between the cortex and the kind of the deeper regions, and then regionally as well. So next we started to investigate, well, the viscoelastic effect. So we started to try and figure, well, if we load these at different rates, so if we do that test fast or slow, and these are kind of on the top, I want to kind of get your attention to the top right corner of the image on the left. You see that in gray matter, as we kind of purple, yellow, and green and blue indicate 25, 100, and 500% per second loading rates. And as we increase that loading rate, there's no real difference in the gray matter. So it doesn't behave any differently if you load it fast or you load it slow in tension. But again, the white matter starts to show this divergence. So the faster that you load it, the stiffer the response. And we're hypothesizing that this could have interesting kind of protective effects, the fact that the gray matter is on the outside. But that needs a lot more work yet. We then took another kind of a batch of samples and we tensioned them to 50% and we compressed them to 50% and we held that strain of 50% for over 2 minutes, and we looked at the stress relaxation, which is another kind of hallmark of viscoelasticity. And again, we're seeing here interesting that there's no statistical significance in the gray matter recovery of the stress, so the stress relaxation, but there is again a statistical significance in the white. So this gray matter is almost like it doesn't matter what speed that you load it at, it starts to kind of it behaves the same, but the white matter then behaves very, very differently in these different configurations. And this is all kind of parallel to the work that's been ongoing with us and Cooper helmets over the last couple of years. And I suppose these are the leading manufacturer in hurling helmets here. And I know it's not the levels of concussion aren't that level of the NFL, but the research is translational across the two in terms of we've still got impact, and we need to maximize that energy dissipation through the layers of the helmet so that it's been dissipated by the time it gets to the skull. So we took these kind of middle layers, the kind of the foam layers, and we started to do these again compression tests, and these are just cyclic tests to try and see, well, if we load it in compression and then we relax it and then load it again and then relax it and load it again, we do 3 cycles and then we leave it for a day and we do the same thing, and then we leave it for another day and we do the same thing. And what we are quantifying here is the area underneath these curves, which is the energy dissipation. So if you look at the red loading curve, which is on top, and then as I relax that material, it starts to decay here, and we call this a hysteresis loop. But this really interesting trend emerged in that the energy dissipated from cycle 1 and day 1 was the highest. And then once you went to cycle 2 and cycle 3, it nearly halved the amount of energy dissipated. So that was basically the first thing. And then we said, okay, we'll look at what happened in day 2, and this recovery happens. So once you leave it for that 24 hours, you regain that energy dissipation again, and then it decays, and then you regain it again. Now, not all the way to 100%, but this, we're kind of, our kind of take-home message from this is that, well, look, the worst thing that can happen is you get these repeated injuries in such a short time frame, so that the energy dissipation is kind of is maximal. So I mentioned the clinical theory of the delamination of the gray and white matter boundary, and that was the next thing that we investigated. So there's kind of 2 ways that these can fracture. There's the mode 1, which is kind of the normal separation, like the kind of the zip effect kind of thing, and then we've got mode 2, which is shear. And we did these experiments and we kind of found these interesting plots. These are traction separation plots, and I want to kind of divert your attention to these 2 green and yellow dots, and these are just the interface strength. And what these 2 dots mean essentially is that there's no difference in a crack wanting to initiate or nucleate in mode 1 or mode 2. So it has no preferential direction to try to start in. But what is interesting is that the fracture energy is different. So you've got this more ductile. Now this is a large strain analysis, so it's a kind of an analogy, but you've got this kind of ductile behavior of the green mode 2 separation, and you've got this kind of brittle fracture of the mode 1. And we're kind of trying to investigate whether at the boundary, whether this is a protective mechanism or not. And the last thing we kind of started to look at in terms of the experimental data was the volumetric effects, or the kind of compressibility effects. And again, we did these experiments and set up 2 different orthogonal cameras, and we're seeing that there is significant compressibility across tension and compression. So there's a lot of, I suppose, experimental data there, and we now need to kind of shift gears a little bit towards how do we computationally model this. And I suppose this really, we used the finite element method, which kind of exploded in the 1960s in terms of the automotive industry started to adopt this. This is a kind of a set of partial differential equations and boundary conditions. They form a boundary value problem, and then we don't have to crash test cars anymore because we can solve the problem numerically. And then this started to kind of branch its way into the biomechanics and biomedical engineering divisions more recently. So we've got these equilibrium equations that need to be solved. We've got our kinematics equations, which relates our displacement to our strain. And then we've got our thermodynamics. So we've got essentially, this is our equilibrium equation. This is our energy, our internal energy, which is equilibrated by the external forces. And we're neglecting dissipation for now. I'm going to come back to that in a minute or two. So the external work is basically kind of an integral over the boundary, or over the entire domain, I should say, of the body forces, things like gravity and electromagnetism and things like that. And these tractions, so these are kind of like the forces when you impact the skull. They're applied to some region of the skull. And then this boundary is our Dirichlet boundary condition, which is basically like what stops that from moving in space. So like when it hits the ground or something like that. And this kind of external force has to be equilibrated by the internal stresses that are generated. And that internal stress is a function of the stored strain energy, which is just a linear elastic formulation for now. And then we have to discretize this in 2D. So obviously the brain is a complex shape, and we can make it up of loads of these, which are called finite elements. We can make up the complex shape, and then we solve this at every integration point in every element. And things like where we can calculate the strain in the element, which is a function of the derivative of the shape functions. We're assuming plane stress, which is a 2D approximation for now, and that's very wrong, but I'll come back to that in a second. The important thing is that we need to solve for this, minimize this residual here. So we need to balance the equations, the external force with the internal stresses. And we can do this using Gaussian integration and solve for this displacement U at the nodes. So we said, great, like we can do this. We came up with a hyperelastic cohesive zone model. And I remember that green and yellow kind of dot that I had said to you earlier, this exactly sits in here. This really gives us the interface strength in mode 1, which is normal, and mode 2, which is shear. And they sit into these equations, and they can degrade the elements, and this kind of zip can open and the crack can form in a cohesive zone model. And we did this, and this is the type of behavior that you see when you subject arteries to when they burst and when they rupture and things like that. And we're able to predict fracture patterns in patient-specific aneurysms and show that these tractions are not coincident with the stresses. So we said, great, we're going to do this. We're going to be the first people to do it for the brain. And then we looked back at our videos and we realized that this is the gray and white matter boundary, and the crack doesn't happen at the boundary. So the brain tissue is easier to break or a crack to form in the bulk tissue than at the boundary. Not in all cases, but in a significant number of cases in both mode 1 and mode 2. And this is where our problem came, in that the cohesive zone model requires a predefined fracture path or a crack path. So let's go back to our drawing board and back to our computational model, and we had to integrate, or we had to, I suppose, add back in that dissipative term. And really, if you think about it, it's a competition now between the surface energy for a crack to grow and the stored strain energy. So if something starts to build up its strain energy, you can relieve that strain energy by unzipping that crack a little bit. So it becomes a competition effect. And we're using the work of Frankfurt and Marigot in 1998, where they proposed a variational reformulation of this. But the problem is still this is an untractable computational scheme, because this crack path is unknown for us. So again, this was replaced by Bourdain in 2000 by the surface integral was replaced by a volume integral, and this becomes a diffuse crack then. Where the intact material, this phi parameter, this is essentially like an additional degree of freedom in the finite element problem. It's equal to 0 in the intact, and it's equal to 1 in the cracked material. And then this phase field damages our tensile part of the strain energy. So we get this kind of softening behavior like we see in the experiments. And then I'm going to fly through this, but in terms of the thermodynamics, we've got this energy functional that we need to minimize and then maintain. This dissipation of energy has to be non-negative. And we get these additional, I suppose, stiffness contributions to the global stiffness matrix, and then we solve for those degrees of freedom. And this is what it looks like. So this is a very simple experiment where we just pull the top and hold the bottom, and you can see the cracks start to zip across. So these elements are initially blue, and then they turn to red as this phase parameter, or the crack, essentially grows. And you can see we grow up here to our interface strength, and then we degrade down in terms of the exact same behavior that we see with our experiments, only that this is a small strain regime. And we can add in more complicated shapes, things like the lateral ventricles. This is obviously idealized for now, but this organic crack branching happens automatically because of the minimization. So just the last slide is in terms of ongoing and future work, and it's really the bulk fracture test, the destructive tests of the isolated gray and white matter. Is the interface protective is still an open question for us. We need to scale this up to a finite strain model. So we're currently looking in the region of like 1% strain. We need to figure out what levels of strain are actually happening in the brain. and then fatigue failure and recovery post-damage. So what happens to that surface? Because the surface has to regrow its strength. It kind of gets damaged, so it decreases and then it recovers again. And then we're using this with the UPMC in Pittsburgh to try and have a mechanistic understanding of when return to play should happen for athletes. So with that, I'd like to thank my collaborators and funding and support, and you for listening. Thanks very much.
Excellent. Thanks a million, Jamie. Okay, so just a reminder as well, if there's any questions that you have, to get them in now, and Jim will be able to answer them at the end. So our next speaker is Dr. Andrew Daly. So Andrew is an associate professor in biomedical engineering at the University of Galway, and a funded investigator at the Research Ireland Centre for Medical Devices. His research focuses on utilising bioprinting technology to create functional human tissues. He holds a BAI and a PhD in mechanical engineering from Trinity College, and after a postdoctoral fellowship at the University of Pennsylvania, Andrew returned to Ireland to establish his independent research group at the University of Galway. Andrew leads a team of 4 postdoctoral researchers and 6 PhD students, and his research has been published in top-tier journals, and he's also secured research funding, including a prestigious European Research Council starting grant in 2022. So yeah, I'll hand it over to yourself, Andrew.
Okay, so thanks, Niamh. Perfect. So hopefully you can hear me and the slides are okay. So yeah, so I'm going to talk about some of our recent work on bioprinting human heart models, and showing how we can use this technique to process stem cells into more functional tissues. So I'll start off with work on bioprinting our heart tissue, and I've also got some kind of more recent work on AI-powered printing, which if I have time, I'll go through. So this is a relatively recent now, 2019 high-profile paper. Sorry, my phone just started going off. From 2019, looking at creating bioprinted constructs mimicking the anatomical geometry of a heart. And so this technique really allows us to print, visually impressive structures using hydrogels, and we can include cells within them. However, if we look at the structure and the organization of printed cells, compare them to real muscle or the adult myocardium, we can see there's major differences. So the printed tissues really lack the structural organization and the alignment, which is essential for electrophysiological and contractile functionality. So there's big challenges to be addressed. 2 ones that we're focused on are how we can really create that very high cell density present in the adult myocardium, and also at a high cell density, how do we create that alignment and anisotropy in the muscle? So we're taking kind of inspiration from embryonic development and how the actual heart forms, which it actually starts off as a simple tubular pump, that over time, due to various types of mechanical forces surrounding it and also growth of the tube itself, will cause this bending and buckling and twisting behavior, kind of forming that early 4-chamber structure. And these are just some images you can see in a developing embryo of the heart tube as it bends. So it's been shown that cells gradually mature during this process, and the forces that are generated within the tissue sculpt cell alignment as the tissue bends and morphs. So we started thinking, if we can incorporate shape morphing behaviors in a printed tissue, could we make it more mature by mimicking these developmental processes? So that's our hypothesis with this first project, that if we print something, say a tube, if we can apply a shape morphing behavior, would the resultant mechanical forces improve the structural or functional maturity by sculpting the cells? So just to kind of break down how we're thinking about tissues, so they're active self-shaping materials. If we have a cluster of cells over time morphing into something more functional, from a physical perspective, we have spatiotemporal variations in mechanics or growth. So we have material addition, which could be cell proliferation. We have forces in that tissue, so that's active cell contractility. So for example, cells pulling on the extracellular matrix. and combinations of material properties too. So stiffness of the matrix will influence how it changes shape. So these three things combine together to give a morphogenetic flow, which then will influence the phenotype of cells. So the first project I'm going to talk about is really focusing on understanding how contractile forces influence morphing in a tissue. So this is work from the first PhD student in my group, Ankita Pramanik, and she developed a printing method here for depositing a collagen bioink that you can see here in yellow. And this is a camera looking at our extruder or printer from the bottom, actually. So we're just printing this simple spiral shape structure. And you can see it's actually been printed into another fluid that kind of holds it in place and gives it nice structural stability. And we can also incorporate cells inside this. So in the schematic, the cells are denoted with this magenta color. And I'll talk about the cells we use in a second. We also add a little bit of hyaluronic acid, which is denoted by this kind of shorter polymer structure. And that increases the viscosity. So it's a nice technique. We can print cells in inks that mimic the extracellular matrix. And this is just an example of, over time, if we actually culture cells in these printed structures. And here you're looking at cells are kind of fluorescently labeled in magenta here. And we've also labeled the collagen with this kind of green stain. So it was interesting to us that if you print something simple like a tube, it will shrink over time. So over 14 days of culture. So that's caused by cells actually pulling on the matrix and creating a smaller construct. If you change the geometry, you kind of get a different shape morphing process. So if we did these M-shaped structures or spirals, we found that due to the cell forces on the ECM and on the tissue, that created a stress that had to be dissipated. So the structure changed shape over time. So it gradually opened, and you can see that here. So this was nice. So we could print something and we could see it evolve over time, kind of within this support bath that I showed before. And we could scale it up. So if we print this tube structure or tubular M structure, the stresses within the tissue during culture would cause it to change shape. So we saw kind of straightening of this bent tube and gradual opening of these M structures. And this is kind of just looking at an angle of them morphing within the support bath. I won't go into too much detail on this. We collaborated with another lab, the McAvoy lab in biomedical engineering at the University of Galway, who are really kind of experts in finite element modeling of cell interactions with ECM. So they've previously developed a model that can account for stress fibers pulling on collagen and kind of the reorganization that happens over time in these tissues. And we were able to create models, these are just snapshot videos of this, that importantly actually predicted some of the experimental results we found. So we saw this kind of isotropic shrinkage of a tube, whereas if we initially had a square shaped structure, we got a different morphing pattern. So the model is quite interesting because if we can predict it before we print, we can then kind of use it to do an inverse design if we have a specific pattern we want to recreate. So I'll go through this quickly, but the other kind of interesting thing that we noticed is we're printing these tissues in a kind of a viscoelastic support. And we found that the viscoelastic properties of that gel that the tissue was printed in also impacted how much it could shrink. So if we had something that was more viscous here, so it's more liquid-like, versus something in the last panel here that's more elastic or has more energy storage, we saw that there was less shrinkage within the tissue, and because it's providing more resistance to the morphing over time. So that'll also have an impact on the process. So we can print something, we can predict it, and it'll morph over time. So why does that matter? So what we actually started seeing, and we wanted to look at, is what's happening to cells within these tissues. So using different types of confocal microscopy, we were able to look closely at the cell organization. And we found, if you look in this geometry, cells being kind of stained in red here, that they took on an orientation mimicking the principal axis of the tissue. So they're kind of circumferentially aligned over time. The model was also able to predict this. So the principal strains that were developing in the tissue or the direction of maximum stretch, cells were aligning in that direction. We were able to predict that with the model system, and also we were also able to kind of mirror similar changes in the extracellular matrix orientation. This is just kind of looking a little bit closer and quantifying it. So here at the start, we don't see too much organization just after printing, but over time we start to see more of it emerging in this particular direction along the principal axis. We really saw it actually in the nascent ECM. So this is matrix that the cells themselves are secreting after culture. So you can see a lot of alignment along the principal axis, and that is an important thing to think about in heart muscle. So how we can get that organization. The previous work I showed you was using fibroblasts as the cell type, which they take up about 20 to 30% of the cell number in the adult heart. But obviously the contractile active cell type are cardiomyocytes. So we worked a lot with iPSCs, or induced pluripotent stem cells, and using various kind of differentiation cocktails, we can turn them into cardiomyocytes. And this is just looking at them spontaneously contracting on the bottom of a dish. So this is like a monolayer of cells. So we can take those cells, kind of process them into a tubular structure that you can see here. So it is contracting in a synchronous manner. So the area is kind of shortening as it contracts. They beat at about 0.5 hertz. If we stimulate them, they will beat a little bit faster. So that's quite promising, obviously. And we saw when we looked at the tissues over time and cultured them for long periods of time, like 28, 50 days and 90 days, if in this top panel, we're looking at structures that shrunk over time when we had a mixture of cardiomyocytes and fibroblasts in the tissue. And we saw, for example, a lot of alignment occurring in that circumferential direction around the tissue. You can really see nice sarcomeres here forming. So those are stained by this alpha actinin or green staining here. And so we saw these tubes shrinking when we added fibroblasts. So we got about a 40% reduction in the diameter in a control kind of static group that was not morphing. And we did this by only encapsulating cardiomyocytes in the tissue. So this had no fibroblasts. And we found that the tissues did not shrink as much, so it was about 10% morphing. And you can really see that had a detrimental effect on the cell alignment. So they're very rounded. The cardiomyocytes are there and they're alive, but they don't have that nice anisotropy that we see in this top panel. And you can see that the sarcomere or the sarcomeric protein is much more disorganized, and that even stayed up to day 90 in culture. So it's nice that we can improve the functionality of the tissue. We can kind of quantify how the intensity of that beating. So we get about a 4 to 5 increase in contractile force when we have this morphing tissue. And again, we kind of did some transcriptional assessment to look at the gene expression profiles. And again, there were upregulation of genes related to ion channels, so that are important for propagating the electrical signal through the tissue. Just maybe to finish, this is kind of a scaled up geometry. So looking at a cross section with two hollow or two structures mimicking a ventricle, that shrunk over 14 days, we can see a lot of a nice troponin staining indicating viable cardiomyocytes. We were able to see some cell alignment along the principal tissue axis, although it's obviously not perfect and there's more work to do. And again, here you can see kind of the contraction of that larger scale tissue. So this is about 7 by 8 millimeters in size. So we're now kind of working to scale this up. I think I have about 4 minutes left. So sorry, 2 minutes left. So yeah, I'm going to talk now just a little bit about another project we're working on, and I'll go through this very quickly. So most printing hardware or bioprinting hardware works quite well, and I'm sure some people are working with 3D printing for their processes. However, in most cases, it's still an open loop technology. It's predetermined. We define extrusion outputs. We give XYZ coordinates and it'll print a structure in 3 dimensions. However, there's no feedback going back to the printer about the outputs. And the printers themselves are not capable really of monitoring process outputs or detecting or correcting bioprinting errors. You can imagine bioinks are quite expensive materials, so when this does happen, it can be quite costly. So we have another project ongoing now where we're trying to monitor the extrusion process in real time. And for example, if we have correct extrusion happening for a period of time, if an error does occur, could we then in real time kind of adjust that and fix it or lower the extrusion rate in this case? So we're developing a system where we observe or watch the printing process using cameras. We're using convolutional neural networks to be able to classify if the state is good or bad or appropriate over time. And say if we've detected an over extrusion, then the plan is, could we implement an immediate correction after that? So very quickly, we've got a printer that has a camera underneath it that allows us to monitor the extrusion process, like you can see here. And I'll just kind of show. Don't have time to go through all of this, but I'll show you the end product. We've basically trained a neural network system that in kind of real time can classify, say, our extrusion as good, and you can see in this case here. So we're indicating that we've got an appropriate extrusion that's defined as kind of the width of the filament. And at that point there, we've introduced an extrusion error. And down here, you can see where we've introduced this kind of closed loop system where if we watch, can we kind of detect if it's gone too far and can we slightly lower it to kind of bring it back to a more appropriate state. If we just compare this image to this image, so this is open loop without our machine learning controls, and this is closed loop with them included, we can see we've actually improved the quality of the print. So we've brought the extrusion width kind of back closer to the nozzle. So we think that's quite powerful for making the processes more reproducible, scalable, and kind of printed parts that are more error-free. So yeah, kind of with that, I'll just thank Daniel and Vasilis who are working on the second project, and Kita who's leading the first project. So yeah, just to thank the funders, and yeah, I'm happy to take any questions if there are any at the end. So thank you.
Amazing. Thanks so much, Andrew. And kudos on the timekeeping. It's not an easy topic to summarize in that time frame. So thanks a million. So yeah, just as Andrew was saying, if you have any questions there, please do pop them in to the Q&A so we can get to them at the end. But yeah, so we have our final speaker for today's webinar. So we have Dr. Ellen Roach. Ellen is an associate professor at the Institute for Medical Engineering and Science and the Department of Mechanical Engineering at the Massachusetts Institute of Technology. Ellen directs the Therapeutic Technology Design and Development Lab, and she completed her PhD at Harvard University School of Engineering and Applied Sciences. Ellen's research focuses on applying innovative technologies to the development of cardiac devices and includes development of devices to repair or augment cardiac function using disruptive approaches such as soft robotics, combination of mechanical actuation with delivery of cell therapy, and use of light-activated biodegradable adhesives. So Ellen has authored over 100 conference and journal papers and also is a recipient of multiple awards, which I have listed here, and it's too many to list off. So yeah, I'll open up the floor to Ellen. Thanks a million.
Thank you. Are you looking at the right screen?
Now we are, and if you want to pop it into presentation.
Let me just go here. Okay, perfect. Does that work, Bob?
Perfect, thanks.
Great, yeah, thanks for having me, and it was great to hear the other talks from Jamie and Andy. So I'll jump right in. I'm going to focus today on augmenting heart function with the device-based approaches, and thanks, Niamh, for the kind introduction. Yeah, I work with a number of companies, so just disclosing them here. My research mainly focuses on cyclical dynamic pumps, muscular pumps like the diaphragm and the heart. Today I'll focus more on the cardiac work. But these muscular pumps obviously are life-sustaining and important for keeping us alive, pumping blood in the case of the heart around the body. So we're interested in how the function of the heart can go wrong and how we can help to correct it. We really, we're a research lab, but I have industry experience, and we partner a lot with industry, and we partner with clinical groups as well to work together to really kind of advance these technologies forward. Recently, our work has focused a lot on bringing together different groups and working on models of disease and diverse phenotypes of different types of cardiac disease. And we do that with benchtop models, computational models, and animal models. And then we use these to develop, test, and optimize devices. So although we do work on designing the devices ourselves, we've really grown the area of like making better test beds so we can, partner with industry that are working on devices. So I'll talk through a few of these test beds today. In the time I have, I'll try to be as good as I'm keeping time and Jamie. Let's see. So the first kind of case study I'll talk about is these patient-specific models of aortic stenosis and diastolic dysfunction. So very kind of briefly, aortic stenosis is when the aortic valve becomes narrowed because of disease, calcification, or because of congenital heart defects, like bicuspid aortic valve. So we gathered with the collaborators at Cleveland Clinic, we gathered a data set from clinical data. So we had CT scans and echo, and we had some left ventricle catheterization. So we had hemodynamic pressure volume loops. So we were able to then segment these CT images to get accurate patient-specific geometry files. And then we use soft 3D printing techniques to actually physically print the left ventricle and the aorta from these patients. And then in the lab, we made these sleeves that go around the ventricle, the pumping chamber of the heart, and around where the valve is located. And they each have pockets that we can inflate and deflate to change both the geometry of the ventricle or the aorta, but also to act as the contractile kind of pump for these models. And these sleeves were also patient-specific according to the patient data that we had. So with this, then we can do both morphologic mimicry, so we can make the valve look similar to the actual clinical files that we have, but we can recreate the pressure waveforms in the aorta, the left ventricle, and we can recreate the stiffness of the ventricle or pumping chamber. And we can use these to mimic disease, but also to understand or study what would happen in this patient if we place a replacement valve, or a TAVR in this case, which is a transcatheter aortic valve replacement. So just to kind of high level show what this looks like, this is the sleeve we made for one patient. So you can see it's kind of designed for their particular anatomy. We put a fabric on the outside and inflatable pockets thermally formed on the inside, and we have kind of a skeleton to keep it in place. When we inflate and deflate these pockets, we get a kind of pumping function of the ventricle, and this is under ultrasound, so we can use ultrasound directly on these models in the lab. And we can also then recreate at the level of the valve, what that anatomy looks like. And we do that with the smaller sleeve that goes around the aorta. So you can see on the top here, these are images from patients, and we can recreate the shape using our sleeve on the bench. We can also use multi-material 3D printing. So we print on calcification onto the valve leaflets. And with this, we can look at things like pressure gradient in the model compared to the pressure gradient we've measured in the patient. And here on the bottom, this is pressure gradient. The patient values are shown in gray, our soft robotic model in blue, and then this multi-material 3D printed model in pink. So you can see both models are pretty good at replicating the actual hemodynamics of the patient, but now it's all on the bench in a synthetic model. Because we're using the sleeve to act as the pump for the heart, We can also change the stiffness by changing the filling pressure of the sleeve. We can change the compliance of the myocardium or the heart muscle, and we can replicate healthy contractile muscle, or we can replicate moderate or severe remodeling when the ventricle starts to stiffen, and this can happen in a specific type of heart failure called HFpEF, or heart failure with preserved ejection fraction. So there's a limited capacity for the heart to fill. This is just a very brief kind of demonstration of what we can use these models for. So this is a transcatheter aortic valve replacement. Before we implant this, and this is all done on a synthetic model on the bench, you can see there's a large pressure gradient between the aorta and the left ventricle. So across that narrowed valve, there's a high pressure gradient. And then we place the valve, and we can see how that pressure gradient really goes down a lot. So the new prosthetic valve bioprosthesis is taking over the function, and it's no longer narrowed or stenosed, so the pressure gradient goes down. And with that, we can look at different valve types and what's suitable for patient-specific anatomies, particularly in cases where they have a very unique or tortuous anatomy. We can also look at positioning and sizing and use these models for training interventionalists and cardiologists. Yeah, this is just to kind of bring your focus to the pressure gradient, which is the difference between these two curves and how it goes down after we implant the TAVR or the bioprosthetic. So yeah, just to summarize that portion, we have these patient-specific models, and we can represent aortic stenosis or narrowing, as well as diastolic or filling dysfunction in the pumping chamber or the ventricle of the heart. This is an advancement upon kind of existing models. We can have enhanced control and recreate both congenital heart defects as well as degenerative diseases that happen with time and age. And we can modulate heart function and then also simulate treatment. This video just in 15 seconds kind of shows the moving model on the bench.
The sleeves could be tuned separately to realistically contract and constrict the printed models. By using patient-specific data, the researchers are able to accurately recreate the same heart pumping pressures and flows that were previously measured in a specific patient.
All right.
The sleeves.
So then we kind of thought this is really nice for a synthetic model, and it works well for the aortic valve narrowing and for diastolic dysfunction. But when we get to a different valve in the heart, the mitral valve, which separates the left atrium and the left ventricle, it's a lot more complex. So we had to think of another way to simulate this on the bench. The mitral valve is a complex kind of valve with cords and papillary muscles and leaflets, and that whole apparatus is very difficult to print or to make synthetically. So we took this other approach where we made these biorobotic cardiovascular simulators. Here we take an explanted real tissue heart, usually a porcine heart, because of the size and it's equivalent to humans. And then we use a very specific process to dissect away the muscle or the myocardium, and we replace it with artificial myocardium. So this is kind of, I suppose, complementary to what Andy presented about printing the myocardium. Here, we're actually making it fully synthetically out of kind of soft robotic structures at a different scale. So, you know, Andy's work is a lot more advanced in actually making the microstructure and the cell structure, the myocardium. This approach, we remove the native and we replace it with a synthetic muscle, as you can see here. This allows us to get kind of human physiological levels of pressure generation and flow. And we can connect it to this system in the lab where we can measure pressure and flow with ultrasonic sensors. We can have cameras in the ventricle to look at valve motion, and we can tune different pressures and the overall kind of preload and afterload in the loop. So this allows us to study a number of devices and obtain real-time hemodynamics and directly observe and visualize, because we perfuse with a clear fluid, what's going on in the valves when you do an intervention. So just to show you this on the bench, this is kind of a biohybrid heart where the tissue is native biological tissue, and we preserve it in a way that maintains the mechanical properties. And these artificial muscles make the heart pump again or bring them back to life. These blue probes here are flow sensors, and they allow us to, there's pressure sensors also within the ventricle, so we can measure the pressure in the left ventricle, the left atrium, and the aorta. And we can also measure the flow across the aortic and mitral valve, and these are quite close to physiological levels. To make this myocardium, we don't just kind of put these artificial muscles wherever we think they'll go. We do a lot of optimization of this artificial myocardium using computational modeling. So this is just kind of showing like the number of different scenarios or placements of muscles and different muscle types that we screened before we built this myocardium. So we can do structural models of different types of this. McKibben is a McKibben actuator, flat or 3D, and there's different types of muscles that we can embed in a synthetic myocardium. And then we can test how much volume they will displace with each contraction and how much rotation, because we want to mimic the twist of the heart as well as the contraction. We can also model how much pressure they'll generate. And then we can pick what's closest to physiological. So in this case, it was this kind of dual layer design with flat McKibben muscles, the muscle type on the outer layer, and then inside some circumferential 3D muscles. So here you can see from the bottom, you get this subapical twisting, and from the side, you get this compression. We can also use fluid structure interaction models to look at how this structural motion will affect fluid inside the chamber of the heart. And you can see in our first design, you get this kind of trapping of fluid, which we really don't want. You get this red represents low velocity, so you get kind of trapping inside. But when you add the twisting actuators, you get like ejection and you don't get any kind of trapped fluid in the myocardium. So this saves us a lot of time in the lab because we can computationally really screen which design is best for this purpose. So going back to the model, this just shows the same thing again, this fluid structure interaction, and we've tuned the model now to get like pressures up to 129 millimeters of mercury and displacement percentages of about 70%, which is great. So if we go back to the physical model now, we have our myocardium designed to get the right properties. And now we can look inside this kind of beating heart, and we can look at the aortic valve opening and closing. This is direct visualization on the left and with ultrasound on the right. You can also look at the mitral valve, and the same thing, you can see the leaflets opening and closing with each cardiac cycle. You can also replicate disease. So here, this is a condition called mitral regurgitation, which is a leaky valve. So the blood flow is going back up to the atrium when it should be going out of the ventricle to the aorta and around the body. We induce this by rupturing deliberately the cords that attach these muscles to the leaflets. And then you get prolapse and floppy leaflets, and you get kind of leaky valves. And you can quantify this. The baseline is shown here, and the more flow you have under the 0 line is regurgitant or backward flow. So moderate and severe mitral regurg are easy to simulate just by cutting more cords. And then we can simulate treatment. So here we put in a surgical valve, and you can see pre-treatment and post-treatment, you know, this leakiness and this kind of regurgitation really goes away when you place a surgical valve, and you get nice coaptation of the leaflets. And you can quantify that too. You can also look at the effect of replacing the mitral valve on the aortic valve. There's also an effect where when it's treated, the aortic valve opens a lot more and can supply more blood around the body. Quantifying that is easy. So you can see baseline and then a lot of regurgitation. And now with the valve, you've got less regurgitation again. And this just shows that the actual regurgitant fraction is restored to baseline after you replace or use a valve to replace it. The final thing I'll talk about is another mitral valve device, and this time developed by Abbott. It's called the MitraClip, and this is actually a clip that clips leaflets of the mitral valve together to prevent this leakiness. And with our model, we were able to simulate this. So do the whole procedure on the bench, insert the MitraClip, position it, and install it, and measure the function afterwards. This is really unique because this procedure is typically done under X-ray, and you have no visualization because the blood it's a beating heart procedure done transcatheter. So this was really interesting for interventionalists to be able to see step by step what they were doing. The video shows leaky mitral function, regurgitation, and then we place the mitral clip, which clips two of the leaflets together, and you can see how it changes the hemodynamics there in the leaflet opening and closing. And we can quantify that again. So we show baseline, moderate mitral regurg, and then mitral clip. And you see how the regurgitant fraction is very much reduced. And you can also look at the effect on left ventricular pressure. So at baseline, you have healthy, and then your pressure goes down, your systolic pressure when you have mitral regurgitation. And once you place the clip, it goes back to baseline. So yeah, hopefully that's a good summary of how we kind of bring together these groups. All these projects require engineers and clinicians and industry sponsors and industry kind of collaborators. And we do benchtop computational animal. We've tried to kind of link them all together to optimize the devices. It takes all those groups to really do it. So yeah, that's it. Thanks to the group here, and thanks to the funders. And I'm happy to take any questions that you